the So this information will be the final chapters. 15 and 16 will be the final chapters for Unit 3, and so then the exam is due by the end of the week, and then we'll kind of move forward with information about the plants. And the first video, or the video lecture about the first chapter in there, which is chapter 6, is already up. But then we'll have some fun with plants and animals as we head in towards the rest of the class. So that's pretty cool. So, time to share. So what we're looking at is a couple of different ideas, drawing in the concept of natural selection and trying to help us understand how that relates to, both Darwin's work as well as some of the other concepts, how they kind of tie together. The important thing is that natural selection itself isn't evolution. Within natural selection, we can see diversity and change within a population, but the key is that it kind of is a vector for evolutionary change over longer periods of time. The main concept, which some people kind of boil down to basically some sort of a survival of the fittest. Basically, fitness in this case is related to simply surviving for an individual within a population to either survive and to reproduce. And whatever sorts of adaptations that individuals have within their genetic code that maybe help them both survive and thrive and reproduce. Well, those are, of course, when they are inherited, those would then be passed on. And so we could see how that particular adaptation might be more and more popular or more and more common within a population. So as the population grows, you can imagine how a trait which might allow for better fitness will itself be passed on and will become a larger and larger portion of the overall population. And this, as it says, will kind of lead to changes in the population over time. It doesn't mean that we're all perfectly adapted. There are, as you learn more and more about the biological world, there are more and more amazing things that you'll hear about in terms of how adaptations are found within different populations, but it doesn't necessarily mean that it is, in fact, a perfect sort of scenario. And the reason is, of course, the environment and nature kind of keeps changing. There's always shifts and changes through small-scale time and through long-scale time with the environment and how adaptations to it it kind of is a moving mark as well. But essentially what we're seeing here is that when we think about genetics and think about the genes in a particular species, it's not like starting from scratch each time. Each time there is sort of an iteration and a variation that leads to new species. But there are other things that need to go into it, and we'll talk about those as well. But it's not like starting from scratch because we have all these different characteristics within. And so a lot of times when people think about, whether we're sort of related to the monkeys, well, actually what we talk about is having a common ancestor, that there was something that an ancestor that we both shared, and that there was some diversion or separation or split. So when you talk about the creation of these species, there is that genetic component. We can look at the genes to see how closely two different species are related, but again, it's not always starting from scratch. But there is also with everything in life, I guess, there is some sort of a trade-off. Every benefit has another side to it. So an example of sort of an imperfection is, walking upright is great because unlike four-legged creatures that use all four of their limbs to help get around, we, on the other hand, are hanging out on our two legs, and that gives us the freedom to move our hands around, to use them for a variety of things, for tools and for typing on the computer and all sorts of things like that. However, walking upright isn't the best thing for our spine. A lot of people have lower back pain or sacral pain in their pelvic girdle or those sorts of things. So there are, it's not like it's perfect. There's always, I'm going to continue walking around upright. I would rather put up with the occasional aches and pains and walk around upright than go around on all fours and not really have the capacity to use these wonderful hands of mine. Okay. So what exactly does occur, or so once we think about natural selection as a vector for evolution, what is it that we're actually looking for? The first thing is variation, that the members of the population differ from one another. So if everything is a clone of each other, if everything has the same genes, that's not going to confer any selective advantage on one individual or the other. Whatever it is, these variations that exist within us, sometimes the advantage of those variations are not immediately apparent, and sometimes it is those variations that actually allow us to allow an individual within a species to become more fit. So there has to be variation and differences throughout a particular population. Another thing is, of course, inheritance. A huge key is that these are characteristics that have to be passed on. Because again, if you don't have the passing on of that, it's not going to become a larger component of that population's overall genetic makeup. And as a result, we're not going to see those shifts. And then, of course, how reproduction occurs, how that is more successful and resulting in well-adapted, successful offspring. So it's not just about whether you yourself, as an individual within a population, reproduce. It's also whether your reproductive output reproduces as well. And when you're putting your kids to bed, I would not recommend just saying, you are my favorite reproductive output. But nevertheless, it's a biological term, not one that engenders sweet dreams by your offspring. Calling them your offspring probably doesn't help either. Anywho, so what does that mean? How can an environment or selective pressures impact the overall variation on a population? Well, most of the things that we're considering, most of the characteristics are in our bodies or in any organism are polygenic, meaning that it isn't just a simple one allele pair that controls a particular trait within us. That is one of the reasons why Mendel got so lucky when he was doing his pea experiments. He had characteristics that were simply a single allele where he could look at the genotype and create the phenotype, and look at or not look at the genotype because he didn't know what was going on inside the plant, but he had some ideas. But he could look at the phenotype and look at these two different variations of plants, and those variations were on a single gene, a single loci, a single allele pair. Most of the time we're looking at things that are far more complicated and that many genes kind of go into the expression of many genes goes into that phenotype on how different organisms look. So a couple of different types of selective pressures would perhaps, if we looked at an initial population like this one right here, where you have from white to yellow in this particular lupin population, you can see that a distribution here in the number of individuals within this population. You have a huge number of pink. In the middle, you have a few white and a few yellow, and that kind of creates this bell curve. And what we're going to look at, this is just as an example of the sorts of things where we see multiple alleles focusing on a single phenotypic look or trait or a single aspect of that population. And we're going to see how different selective forces might have some differences or might have some impact or selection on it. So the first type is called directional selection. And this occurs when one of the phenotypes at the edges of the bell curve that we see in a population, one aspect of it is favored. And so what's going to happen in this example, larger body size, I guess we had these tiny little horses, and in this case, the overall population of these cute little teeny horses prancing around, going to the rodeo or whatnot, we have this large diversity of body size, but in this case, we're imagining a situation where the selective pressures are for larger horses within that particular species. And so what we can imagine is if there's a selective pressure that actually selects a larger body size in this particular animal, this diversity exists. There's still large members of this little teeny horse population. And so what can happen is it shifts, and after time, larger and larger percentage of the population. If people are like, if the other teeny horses are like, damn, I like these big old, I'd like to have some reproduction with a larger horse within our population. If there's some advantage to that, to their fitness, you can imagine then after time the population will actually grow until you have, in this case, a much larger population. The same genes that express the body size are there. It's just a much larger component of the population. And in this case, through time and through separation, we actually have a shift. That shift is actually continued amongst the body sizes essentially until there's actually these are different species. We have separation of those, and so eventually you have a portion or a population of larger horses that actually are separate from that either have a reproductive separation or time or some other separation from the teeny horses. So these medium-sized horses succeed. Say there was a flood and I'm trying to be mean and kill all the little teeny horses, but say there was some sort of a flood and all the teeny horses died out, but the larger ones succeeded. Eventually, through time, there was that separation that would allow them to sort of survive and thrive. And continuing this directional selection, in this case, after more time still, within the overall diversity of sizes within the Merychippus, this intermediate-sized horse, we actually have selective forces that lead to Equus, the horse that we see today. Everyone okay? No one's like calling Pete about the little horses or anything? Okay. Oh. So, what are some other examples of this? Of course, Equus adapting, because essentially these were animals. The first horses were animals that lived in more of a forested environment, and there was a lot of protection amongst the shrubbery and things like that. But as they grew, being larger, and through time, these areas became more populated with more grasslands, and the larger horses were actually more successful. And so through evolutionary time, we can see how the larger portions of the smaller species were continually selected for, ultimately resulting in the horses that we see today. Another example would be pesticide resistance or drug resistance. Oops. So in this case, directional selection, ignore the horse on the right there, but essentially you have the distribution of a particular bacteria or an insect, and a relatively few have the characteristic of a pesticide resistance or a drug resistance. The funny thing is that might be energetically expensive. And so in a world without pesticides or drugs, that is not very common within the population. It exists there, but until they actually are exposed to the pesticides or the drugs that actually shift and result in a small portion of the overall variation within that bacterial or insect population will shift, and greater and greater portion of them will actually have that resistance to either a drug or a pesticide. And those are good examples of how these things perhaps exist. It's not like, they're somehow like, well, we knew there was going to be this drug and they knew that it was going to, there's no knowledge. It is simply the creation of a new environment, drugs in the body, and how bacteria resist those drugs, or pesticides, and how a population, an individual can't just sort of say, oh, you sprayed that pesticide on me. If they don't express those genes that actually have whatever resistance is conferred, they will still die. But the overall population, there are some members that are going to have that resistance, and they, of course, that's going to help on their selection. All those dead bugs, not doing so well. But the insects that actually have that resistance will, of course, survive. They'll reproduce, and they'll pass that on to their offspring. And suddenly you have a shift, a directional shift, to more resistance in this case. A second type of selection or selective forces are kind of when you have selective forces on either end. Essentially what it is saying is the intermediate phenotype, and when we're looking at this variation, this intermediate phenotype will actually be favored. And so the extremes on either end will not. This example right here are these Swiss starlings. And their clutch size, that is the number of eggs that they lay. In the middle here, it's sort of 4 to 5 eggs. Fewer, some of them have less than 4, and some of them have more than 5. Well, as it happens, this stabilizing selection, 4 to 5 eggs seems to be sort of the best option. The ones that have fewer than 4 to 5 eggs, they don't have enough offspring to actually succeed. And the larger ones are spending so much energy on reproduction and creating more eggs that is an energetic cost that makes them less fit. So in this case, there's pressure from both directions so that you can see through time, after more time, you can see the stabilizing the intermediate phenotype. In this case, those that do 4 or 5 eggs are sort of the most successful and the largest proportion of the overall population. You'll still have a couple of those nutty starlings who are like, I will have 8 eggs, woo! And some that have 2 or 3. There still remains that genetic variation. But because those that do 4 or 5 tend to be the most successful, that will become a larger and larger portion of the population. And so this is known as stabilizing selection, whereas directional selection, you can see it going in a particular direction, looking at a phenotypic variation. In this case, along the phenotypic variation, you have a greater and greater proportion through time in this centermost section. So the intermediate is favored. The ones on the end, the extreme phenotypes, are less favored, and you have this situation where, again, a larger and larger portion of the population has that characteristic. So we talked about directional. We talked about where it goes in one direction or the other. We talked about stabilizing, where the intermediate is more successful. Next, we're going to kind of talk about disruptive selection. And with this case, what we see is that the two different extremes are actually favored. And so there is some disadvantage to being an intermediate phenotype and instead being on either end. A good example of this are everyone's friend, the British land snails, who are found in a variety of different habitats. So you can imagine, in this case, in terms of the banding pattern, you have some that are very low, very, very low banding, and some that are very, very high banding. And those that are intermediate are selected against. And here's why. Because when you're in a field, something like this with this firm brown color, anything that lacks the light bands, something like this is going to survive in the fields because it kind of blends in with the variations in color in the grasses. Well, then those particular snails will do quite successfully, whereas these ones here, the more solid color ones, will kind of stand out against the variation in the fields. In the forests, however, it's a little bit darker, not as much sun gets to the ground, and so the brown ones are going to survive because they kind of blend in with the ground. And these ones that are lighter will actually pop out against the ground, against the soil. And so the thrushes, the birds that eat these, will very much eat these ones at this extreme, and whereas these will survive. So if we look at this species over time, you really don't have, essentially, if you have intermediate banding, you're not going to do well in the forest because you're still going to pop out against the ground, and the thrushes are going to eat you, and you're not going to necessarily have that level of camouflage that one with a lot of banding would have, and so you might not blend in with the grasses quite so much either. And so ultimately what you're left with are sort of these two populations the same species, but one that has a very low banding pattern and one with a very high banding pattern. So imagine the initial example where you have sort of this diversity, but the pressure is against those individuals in the middle, and ultimately you have this disruptive selection that separates into these two different phenotypes. Now, something I wanted to point out each time that we were looking at those examples is to try to understand that variation still exists within the population. There are still going to be a little bit of all the different kinds, even if one or another of the phenotypes are selected for. There is still the maintenance and maintaining of that genetic variability and those genotypic variations within the population overall. You can imagine if you think about traits that exist, just some of the inheritance and the Mendelian inheritance that we talked about, these things often will remain. Dominant characteristics don't dominate the entire population, and recessive characteristics can be quite common. So you have that maintenance of genotypic variation, even though there are forces that are acting on that. The other thing is, and this is very important because it's underlined, is that forces that promote variation, there are other things that lead to variation, are constantly part of the pressures on an organism and on a population of organisms. So you're always going to have recombination. In meiosis, you're always going to have recombination. You're always going to have genetic, every reproductive event combines the genetic material of two individuals, and which genes of that individual, variation according to independent assortment, all these sorts of things, and of course mutations themselves always are going to lead to more variation within the population, in spite of, in addition to the naturally selective forces which may select for particular characteristics. Something called gene flow is also very important. With gene flow, what we're looking at is when new individuals join a population from, say, somewhere else, and they will actually that movement of genes, the movement of genes within that population, when new individuals either leave or join a population, can actually lead to, of course, variation as well. So, at the end of the day, natural selection will favor certain phenotypes, but the others will, as I've said, remain within the population. Of course, something like a natural disaster tends to be a sudden thing and an extreme thing, and of course, randomly happen as well. So, kind of when we're thinking about the environment, there is perhaps more, there is a lot of sort of slow consistency to its change, but extreme events can, of course, happen at random. And perhaps some of you are thinking about those teeny horses drowning. That's perhaps an example of that. There is certain advantages to being heterozygous. Because the advantage of the heterozygote is that it still retains that genetic variation because there are two different alleles. Being heterozygous means that you have both the expressed dominant trait and the recessive unexpressed trait. But only the particular characteristics that are expressed would and are part of your phenotype, part of what you look like, will be actually subject to naturally selected forces. The other gene will remain within you, but is not expressed. But if the dominant gene that is expressed allows you for better fitness and allows you to be naturally selected for, because within your population, that other trait gets to kind of come along for the ride. Down the line, there may be a shift in pressures so that recessive allele within your body or within your genetic genotype that's perhaps passed on to others might result in greater fitness as well. An example of this is a very common disease in Africa, as well as in African Americans, as well as the Black community at large, known as sickle cell disease. Essentially, this is essentially, if we're looking at this particular disease, it's a variation. You can see it's quite a complicated. It's not just one letter. It's a lot. There's basically three different components to this allele. There's the recessive, the dominant, and then there is the A here. Individuals, and HB actually stands for hemoglobin. Seems like a strange sort of shortening from it, but the hemoglobin would either be normal or will be sickled, and the sickle-shaped hemoglobin will result in cells in the body that, under certain conditions, will actually shift to a sickle cell shape. So kind of like a little flat curved, like a flat curved frisbee. So we've all seen sort of images of red blood cells as being kind of like a nice big healthy inner tube. Well, a sickle cell actually kind of is flattened and has a curve to it. And the danger of that is that whereas our sort of, if you can imagine like our bloodstream being being a lazy river, and our blood cells being all these sort of inner tubes, they kind of bounce up against each other and they just kind of flow down the river. With a sickle cell, what can happen is during what's called a crisis, those cells can actually kind of get caught, and because of their shape, they can actually kind of cause basically problems in flow. Now, you may wonder, well, with that situation, someone who is homozygous for the recessive sickle cell trait will tend to die early. However, individuals that have the sickle cell trait are heterozygous and have both the dominant normal blood cells hemoglobin and a sickle cell will still have the sickle cell disease. They will only happen at low oxygen concentrations, and so a crisis would only occur less often. The other interesting thing about individuals that are heterozygous for the sickle cell trait is that malarial parasites cannot live in those that have heterozygous have heterozygous blood. So it's a situation, if we think about malaria spread by the Anopheles mosquito, it's injected by the mosquito into our blood. The malarial parasites live. It can be quite a damaging thing. DDT. I mean, and this wasn't just something that happened in Africa. Malaria, it was a worldwide issue. Back before they started draining things in Washington, D.C., it was known as the malarial swamp. When they selected it for as the capital to our nation, it wasn't just sort of commenting about the bad stuff happening there. It was quite literally a malarial swamp that needed to be drained because there were Anopheles mosquitoes there. Malaria continued to be a problem in the United States and worldwide until the discovery of DDT, which is a pesticide. And if you've perhaps heard about DDT, you may have heard of Rachel Carson, the famous environmentalist, who kind of discovered that DDT was impacting a lot of eggs from things like bald eagles and pelicans and a lot of animals that eat fish. What they found was that DDT was detrimental to the birds who were unable to create more eggs. Now, DDT didn't really have a damage to humans, so it was, on the one hand, it was being hailed as this chemical thing that was helping with malaria. I think I've gone a little too far off the end. It's very interesting stuff. I can obviously talk about it for days and weeks on end. Point here, though, is that if you actually have the sickle cell trait but are heterozygous for it, you actually have less chance of sickling, less chance of a crisis, and malaria cannot actually you can't get malaria. You're immune from malaria because of some kind of coincidence, some interesting thing. And so as a result, this is one of the key reasons that the heterozygous condition and that the sickle cell gene actually remained within the populations, because it conferred, when it's heterozygous, it actually conferred an advantage that neither the homozygous for the sickle cell, which again would die early, or the heterozygous who actually had the normal blood cells would not have. So it actually was an advantage in that case. So here we can see areas that malaria is found, of course, throughout. Continues to be a problem. Sickle cell disease, we can see some of the locations. And then here we can see situations in purple all along equatorial and lower Central Africa, as well as some of these areas along the Mediterranean and into India, areas that have both malaria and sickle cell. And if you can imagine, again, this is an example of the heterozygous advantage. Someone with the normal genotype, someone who is a carrier but has protection from both, as well as the individual with two that is homozygous for the sickle cell trait, who will actually die. So you think, why on earth would the sickle cell trait actually remain in the population? Wouldn't it kind of kill out itself? Well, no, it won't, because as it turns out, it confers this other advantage. So the first thing we're going to talk about is something called microevolution. This is sort of evolution or change on a small scale. In the second part of our lecture, we'll talk about macroevolution and what happens at larger scale species changes and the like. Important thing. This should be underlined 3 times. Individuals do not evolve. Evolution occurs, and it is phenotypic and genotypic changes within a population. All the members of a single species. If we go back to that sort of hierarchy of biological study, Population is a group of organisms in a particular species. So when we think about microevolution, we're thinking about small changes that occur from generation to generation or through a shorter period of time. Again, thinking about how these changes could ultimately lead to things like speciation and the like. Again, these are the same sorts of rules that we've been seeing for natural selection. Genes that have more than one allele. You can have shifting of alleles during sexual reproduction, creating a range of phenotypes within that population. So when we talk about a particular population, in terms of the genotypes of the population, we refer to that as the gene pool. You've perhaps heard that term. Everyone has probably made some joke about some person who should get out of the gene pool or whatever, essentially suggesting that, we as a humanity have a collection of diversity, and some people do not belong. That's not my that's not my cup of tea, but I know that probably is where most of us first hear the gene pool. But essentially what they're looking at is all the genotypes within a particular species. And the example that we're going to talk about is the gene pool of a population of peppered moths. Yes? Oh. Yes, is there a question?
Yes.
Okay. Okay, so we're going to talk about an example of the peppered moth. So the peppered moth is a moth that has basically 3 different genotypes: the homozygous dominant, heterozygous, or homozygous recessive. The dominant condition here is the dark coloration. The recessive condition, found only in the homozygous recessive condition, the only time the recessive gene is expressed is when you have the sort of the peppery color or lighter color of that. And in this example, you can actually calculate within a population based on the number of homozygous, heterozygous, and recessive. Based on the number of homozygous, heterozygous, and homozygous recessive individuals within a population, you can actually calculate the overall amount of genotype, the frequencies of genotype within that population. And so you can look at all of the individuals and determine what percentage of the population is either homozygous dominant, heterozygous, or homozygous recessive. And in this case, we're looking at an example where 64% of the population is this white coloration. So a larger portion of the population is in fact homozygous recessive. About 32% of the population is heterozygous, and about 0.4% of the population is homozygous dominant. So, what we then do is we can basically determine, based on these genotypes, how common the recessive gene is within the population and how common the dominant gene is within the population. So in this case, we know that all of the genetic material from this 4% will actually have the dominant gene. All 64% of the population, all of their genes will be a little d. And if we can imagine this 32%, we can divide so that 16% are large d and 16% of the overall genetic material in that pool are little d. And so when we look at the allele and gamete frequencies, we can see that 4% from here, half or 16% from here, 4 plus 16 is 20. 20% of the overall genes within the population are big D, and 64 plus the other 16% of this heterozygous resulting 80%. So we've taken this population and broken it apart based on the genotypic frequencies to determine what percentage of the overall genetic pool is recessive genes and what percentage is dominant genes. And what we can then do is actually sort of look at that from a perspective of a Punnett square. When we have 20% of the population having a large D or the dominant trait, 80% of the population having the recessive trait, we can actually take that population and essentially cross it with each other, cross it with itself. And so we can imagine that, assuming that there are equal proportions of males and females, 20% of the eggs are going to have, or 0.2, are going to have the large D, 80% are going to have the recessive allele, and 20% of the sperm are going to have the large, and 80% are going to have the small. And look what happens when we actually cross. When we did a monohybrid cross like this, we just sort of did, this one's big D, little d, big D, little d, big D, and little d, little d. Well, what we're actually doing is crossing the population with itself and using the proportions of those genes within the population and actually multiplying it out. And look what we got. 4% is going to be big D, 64% is going to be little d, and we're going to have the other 32% is going to be the heterozygous condition. So if you work it on out, you'll actually notice that that is the same proportions, wrong way, Roger, that we saw originally within the population. So when we're crossing a population with itself, using this Hardy-Weinberg situation, it actually remains the same, where we have 4% are going to be heterozygous dominant, 64% are going to be 4% are going to be homozygous dominant, 64% are going to be homozygous recessive, 32% are going to be heterozygous. And so what we do is we kind of calculate P is the frequency of the dominant allele, Q is the frequency of the recessive allele, and the Hardy-Weinberg equation is P squared plus 2PQ plus Q squared equals 1. Great. What does that mean? Well, essentially, this is, first of all, whenever you asked, who's ever going to use FOIL, first, inner, outer, last, in, maybe that's something they don't teach anymore, but when you're looking at quadratic equations in math class, this is your answer. In biology class, in the 5th week, you are going to be using quadratic equations. Now, I don't want anyone with who's not fond of the maths to run and hide, because it really kind of simplifies it. If we're actually looking at this quadratic equation, we're basically multiplying d plus big D plus little d times big D plus little d. If you think about it, p squared is going to be homozygous dominant. Q squared is going to be homozygous recessive. And the 2pq is here and here. That is the heterozygous condition. And we have 2pq, so this is our p squared, this is our q squared, and these two boxes are our 2pq. So, just something to kind of mull over and think about. Tried to present it in a way that sort of makes a little bit of sense. At the end of the day, we always assume that there are two alleles, and therefore the p plus q equals 1. And of course, if you put that parentheses, p plus q times p plus q equals 1 as well. That's what that is expressing. So it's there. Spend some time with it. We can revisit it in lab tomorrow if you need to kind of think a little bit more about it. So this creates an equilibrium. If you remember, when we started this example, we had a population that had the 4%, 32%, and 64%. And that remains the 4%, 32%, 64%. 4%, 32%, 64%. That remains within the population in perpetuity. This is an example of a population that is very stable. So there are assumptions to the stability of allele presence within a population. First of all, you can't have any mutations, because a mutation would interrupt this perfect ability to kind of maintain these levels of these alleles within the population. We cannot have any gene flow. We can't have anyone leave, and we can't have any new individuals come in. It is a specific, isolated population. Mating is random. Good luck with that. That's not really, anyway, no genetic drift, no selection. What is this saying? This is basically saying is a hardy Weinberg can exist, but it is in the absence of any of the things that we think about when we look at natural selection, when we look at evolution, when we look at changing or a micro-evolving organism or population. So these things are not things that, basically what it's saying is microevolution is occurring and can be measured. And this is where we bring out the example. This is why we talk about these peppered moths. You just thought we loved moths. But no. A really, really interesting, famous study of populations of peppered moths in England. So, a long time ago, in a land far away, or Britain, as the case may be, there were peppered moths. And there were some light pepper moths, and there were some dark peppered moths. And they all liked to hang out on these birch trees. And as it turns out, the white peppered moths with the recessive traits did quite well because the birds had a difficulty seeing them. These darker ones, however, would stick out like a sore blackened thumb against the birch trees, and the birds would eat them. And so as a result, there were pressures against the darker moths, and so the lighter moths took up a larger and larger portion of the population. Now, it is more than likely that still some of these existed. This was the dominant trait, and it is worth within the population. Maybe some of these go visit other trees. Now, what happened was during this time, during the late 1800s, Industrial Revolution, the advent of the discovery and use of coal to really get that industrial revolution going. And one of the side effects of that is that nearby the trees, nearby the coal burning factories, soot would actually occur on those trees. And so as a result, the soot on those trees took these lighter colored trees and darkened them. And so what happened was suddenly the selective pressure shifted. Suddenly the darker moths blended in quite nicely, while the lighter peppered moths stood out like a white thumb against this background. And so as a result, now the birds were the visual predators. It was easier for them to see the white peppered moths, and so the pressure against these pepper moths probably shifted so that they were no longer 64% of the population. There was a microevolution, a change, and more of the genetic success would be amongst the darker moths. Now, again, you can kind of think about through time there was regulations about how coal was utilized, as well as how much they could kind of spew their soot into the air. And so you can imagine then with time, these darkened trees would actually lighten up. And because a lot of these individuals were probably heterozygous, there was still the genetic information for there to be white moths. And so as the trees recovered, you can imagine the shift in selective pressures once again occurred, where as the trees were lighter and lighter, the lighter moths actually succeeded. And again, there was a shift within the population. So these are examples of how natural selection can shift over time and have an impact on a population, in this case, in different directions, based upon the environment around them. So what are the causes of microevolution? Essentially, these are the things that we talked about that are not everything that Hardy-Weinberg and the Hardy-Weinberg equilibrium kind of hates. So, what are they? Genetic mutation. Again, a key way to get new genetic information is if there is a mutation. And sometimes that mutation really helps out and allows that particular individual to succeed. A lot of times when we think about mutation, it kind of has a negative connotation, but it can in fact help individuals succeed. Gene flow, what we're talking about. Gene flow was flow into or out of a population. When we have new genes coming in, or some of the members leave a particular population, that can result in a flow. Non-random mating, as anyone would perhaps, anyone who's dealt with mating, it is not a random thing in in humans, in pepper moths, and anywhere else. Genetic drift. Now, we didn't talk about this that much, but genetic drift is a situation where when you have a very small population, you can actually lose some of the genetic information. Genetic drift is also sometimes called like a bottleneck. So a bottleneck is when you sort of have a larger population that suddenly gets smaller, and when that population gets smaller, there's less genetic information. You basically have a shrinking of the genetic pool. This is something that we deal with a lot when we're talking about endangered species. If there are only 17 black rhinoceroses in the world, this was once a powerful, large population of black rhinos. Hopefully there are more than 17, but there could very well be less than 100, to be honest. And zoos are often doing a lot to help maintain the variation and maintain the number of individuals on these populations, these specific organisms that are so incredibly endangered. Well, what happens is those few individuals, say there were 10,000 black rhinoceros at one point, well, you can imagine that 10,000 are going to have a huge amount of variation within them. When you only have a very, very small population like we do now, you're not going to have that you're not going to capture all of that genetic variation in just a couple of dozen animals. And so that is what's called a bottleneck, and that was what leads to this idea of genetic drift. So you need to have a large enough population to maintain that. But genetic drift is a key way that microevolution occurs. And then, of course, natural selection as well, which we've been talking about. So let's talk about them a little bit more. So genetic mutations are a source for new variations and may have some form of adaptive variation. And I was trying to coach this so you didn't go out and visit your next freckled friend and say, just point at them and just yell mutant. Hopefully with the X-Men, we've developed a way to talk about mutants. Yes, they're superheroes. Maybe that's sort of sending shifting it a little bit too far in a different direction. But mutations can be adaptive. And a good example of this are freckles, which provide more melanin for individuals. Yes, individuals with freckles can burn, but when you compare them to, say, an individual with a very, very light or a non-melanous skin, their risk of things like skin cancer, the risk of burning, and the risk of skin cancer and whatnot raise considerably. So if you imagine, of course, when we're talking about the diversity of skin color, which is sort of, there's like at least somewhere between 8 and 64 different genes that can lead to variations in our skin color in terms of humanity. You can imagine sort of your Tilda Swindons, maybe she could use a few freckles. Maybe that would help her in terms of existing in the environment that has a hot sun with a lot of UV light that can cause those sorts of things. Number 2, gene flow, also called gene migration, as is suggested, things moving into and out of a particular group, and that's separate. Down the line, we could actually, if you have gene flow, between two populations that were initially separated, what you would have is you'd have sort of the accumulated pool of the two groups of individuals of that species. Now, if they were separated, as we'll see in the later half of this class, they would actually maybe have a little bit more variation and perhaps ultimately lead to speciation. But that's not the case. But if there is gene flow between them, they would actually prevent new species from forming. So again, this particular example shows a particular species of snake that has variation, but each of them kind of represents a different subspecies of snake. So they're all potentially related. They're all essentially the same species, but in this case, they have, in addition to the species, or the genus and species, they actually have a subspecies name. And each of them kind of lives in particular environments, maybe the sandy areas of eastern Texas and Louisiana. This one that lives along the Mississippi and some of the hotter areas there. A small batch living right here in southern Florida, and the like. And you can imagine if these subspecies interacted, and if there could be flow between these different groupings within the species. But because they have isolation, they are separate from each other. Now, they're not so separate that they're different species. In fact, if you put one of these guys from East Texas and one of these guys from coastal South Carolina in the same room, they could still mate. They're still the same species, but they have had separation because of their location, because of their environment, because of their habitat. They're each adapted for their own unique area, but they have not yet reached species separation yet. Maybe they will at some point in history, in their in geologic history, but that has not quite happened yet. But you can again imagine how maybe snakes in this small area might interact. These two might interact, but it's unlikely that one of these guys is going to actually, unless again you got a little snake hotel for them, got them a room, etc., where they wouldn't necessarily interact. Non-random mating, of course, like I said, a reality of life. The selection of mate does not happen simply by chance. There is either genotypic or phenotypic reasons for why someone selects a particular mate. This may, if you've ever perhaps offered yourself as a individual for non-random mating, let's just say, and they politely decline, you can simply not take it personally, but know that you're just not their phenotype. So it happens. Anyway, particular character, so there's a sort of mating where individuals of the same phenotype tend to mate. That's not just true in the human world. That's actually true across a lot of organisms and across genetic, with some sort of genetic consistency as well. someone who is homozygous actually may be more likely to mate with someone who is homozygous than someone who's heterozygous or whatnot. And in terms of sexual selection, there are particular characteristics that might increase the likelihood of mating. So, for instance, a male peacock that has a more substantial collection of plumage might make them more give them advantages in terms of sexual selection than to someone with a male peacock that has like 3 sad little plumes and is trying to shake them around and doing it the best with what it has. There might not necessarily be a genotype for that. I mean, they all have similar variations, but there is within that individual there is perhaps the rich, large plumage may be more attractive to a female looking for mates. Talk briefly about genetic drift. We'll kind of genetic drift, as I said before, is what happens when there is a smaller population. Changes in the changes that may happen to chance. There might be a smaller amount of diversity within the genetic pool of these smaller populations, resulting in drift, depending upon who survives, who reproduces, and the like. Talk briefly about the founder effect, the bottleneck effect, where you have a large population that suddenly gets smaller, like we're dealing with a number of different endangered species. The founder effect might be when a new group becomes a founder or actually creates a new population in a new area. If there is separation from the old population, this new population again might not have the entire genetic information within that population reflected in that small group creating. So those are examples of genetic drift. So in this case, you can imagine how, as this we have this population, some sort of natural disaster occurs, killing 5 frogs, and the result, if you look at the overall population, this brown frog is only the phenotypically brown frog is only 10% of the population in this initial, but then once you have the situation where 5 of these frogs are killed in a natural disaster, suddenly they are a larger percentage of the overall population. And in terms of mating and in terms of expressing those genes in offspring, it's more likely that they're going to kind of that the brown coloration is going to be a larger part of the overall population. Just because, again, you've sort of had this shift from a larger population to a smaller one. So it drifts from less to more of the overall population. Thinking again of a bottleneck, quite literally, they like to demonstrate this with a bottle, with a neck, and you have this population expressing a variety of different alleles, and then you sort of pour some of them out through the bottleneck, and you have a population here that only has certain components of the alleles. And you can see, first of all, that there are no yellow balls that have gotten through this bottleneck. So that impacts the overall genetic diversity within a population. You can see that in this case, there are a larger percentage of green in the final population than the initial grouping, maybe fewer blue. So we see the drifting over time of the genes based upon sort of these impacts. So in this case, the bottleneck. Yes. Nope. Okay. So in this case, the what remains in the bottle is sort of all the ones that have died. And again, like the frogs, we can see a shift in the population diversity and the population phenotypes, where only a small remnant population remains. When that population rebuilds, you can see a very different looking population than what started. So there is that loss through that bottleneck. When you have a founder effect, that's where, when you have only a few individuals reproducing, oftentimes rare alleles will be more common in a higher frequency when the population is isolated or separated. A lot of times, you may have actually read an example of someone with double eyebrows. I think that was one of your pedigree questions from last week's lab. That is actually a thing, and there's this island off the coast of Ireland where you have the double eyelashes. You actually have a second set of eyelashes underneath, and that is a gene that is rare, but a population kind of went over there. It was a small group, and as they interbred, that particular trait kind of popped up more. If you have something that's rare, and you know the possibility of it being expressed, especially if it's a rare recessive trait, the possibility of it being expressed is more likely if you have sort of a smaller population. This is one of the reasons why, we were talking about a little bit in lab, like why in these pedigrees did they keep mating with their cousins and the like? Well, that's an example of why, because within your family, the likelihood that you have we all have these rare, interesting, odd things in our genes that never get expressed because they're recessive, and usually we kind of look for mating outside of our families, and in doing so, the other individual has weird genetic things about them that you don't have. So if my weird recessive traits and my mate's weird recessive traits are different, usually the other one kind of has the dominant trait to not express it. But if you start treating family functions like a speed dating service, suddenly the likelihood within your genetic code that someone closely related to you is might in fact have the same weird genetic stuff that you have, and that is more likely to be expressed, especially in small populations. Or in this case, what I'm trying to talk about is sort of mating within family. The same sort of idea is these rare alleles will be a higher frequency in a smaller population that's isolated. And if you look generation after generation, the possibility of them expressing themselves is far higher. This is why in most cases it is illegal to marry someone other than the closest relative would be a first cousin. But again, maybe find someone from a different country or a different part of the country or something like that. Just putting that out there. Okay. So that's the end of the material from that chapter. So we'll kind of move through this a little bit quicker and try to help talk a little bit about speciation and finish up the material for chapter for Unit 3. Okay. So we talked about all these different things happening to the genetic code, happening to that. Now we wanted to kind of take a larger look at how evolution, as we kind of commonly think about it, occurs, and what are some of the various different pieces of evidence and other things that we have that really kind of help us understand the world around us. So we're looking at things like macroevolution and speciation, actually creating new species. When there is a split and that separation, and those two separate new species kind of adapt to their environment and shift and change, and of course, then sometimes we actually see extinction as well. So what are the things we look at? We look at fossils. Fossils help us understand if we want to learn the story of horses and horse evolution, from the little tiny horses to the grand majestic creatures we have today, we can look at the fossil record. We can actually look and see the teeny horses, the medium-sized horses, and the like, and think about, as we were talking about last time, where in the strata they were, just like Darwin did, but we've had a couple centuries now to kind of search to understand the fossils and whatnot. Of course, for fossils, it needs sort of hard parts. It's rare, it's harder to imagine. In this case, we have a trilobite, which was an arthropod. So this was like a big old insect creeping and crawling all over the place, actually in the water. But it's probably closer to like some weird. Anyway, it's a trilobite, and this is the hard shell and the exoskeleton of this particular animal has kind of remained and allowed us to sort of see when minerals replace the structures of it. Of course, plants have left a whole lot of fossils as well. The DNA of every organism. Looking at the DNA sequences, we talked about that process, why we learned the human genetic code, as well as learning about all the other sorts of organisms, plants and animals. It's something that we all share. All the stuff we're talking about with protein synthesis and transcription and translation and replication of DNA, all that stuff is shared across the entirety of the biological world. So what does it mean to be a species? Well, number one, interbreeding. I mentioned that a little bit in terms of how species breed with each other within a species, but there is separation of species because they do not sort of interbreed. They share a particular gene pool, and again, they're reproductively isolated from other species. Sometimes there's a variety of different mechanisms by which a species would become reproductively isolated. It's not just based on how it looks. There are some people who study mosses, and they can look at all these different mosses, and they know how they're different species because of genetics or because of things that you or I, when we're looking, we're just like, it's a moss. No, there are thousands of different types of mosses, and they're all sort of different in their own unique ways. That being said, amongst all biologists, most people are the weirdest, but that's a different issue. Fungi people are almost. Anyway, flow between populations occurs of a species, but not between species. So if we think about those snakes we saw a couple slides back, there could still be gene flow between those populations, between those subspecies. They're all still related. There can still be reproduction between different members of that same species group. So, of course, the species is below the rank of genus. In a genus, you will have one or more species. We can think of the genus of people is Homo, and sapiens, wise people, is the genus and species. We perhaps have an earlier relative, Homo habilis, Homo erectus, a variety of different species within the Homo genus. And what that does is it basically presents the information that there is a common ancestor, that there is relationships, very close relationships. It's the closest two species can have to each other is if they're in the same genus. So again, these are three different species, and a bird person would basically, they would know these differences. They would hear their songs. There would be so much that they know about these three different species. To us, to most of us, we kind of look at them and be like, yeah, it's the same bird. I mean, they just took a picture of the same bird 3 times. This is a ripoff. No, these are actually 3 different species. And they're all in the same genus. They're all in the Epidonax, but they all have their own characteristic species name, their species epithet, and they are, in fact, different species. The book did not rip you off. Humans. Any human can interbreed. We're all the same species. Think about dogs. I don't know if I'd like to watch a Chihuahua and a Great Dane breed, but I guess it's possible. They're still all in the Canis familiaris species. So what is it that can lead to, when we have populations that are closely related, what is it that can actually lead to a species? What can lead to the prevention? That first slide was very heavy on the reproductive isolation and separation of those 2 species. Well, in order to do that, you need to necessarily have something that stands between 2 individuals of a population that are close but not related, not the same species. What is it that we actually have to create that separation? Sometimes that occurs before the mating. So their separation, they may live in a different location. They may live at different times. They may be active during different times of day. They may have different behaviors. And those sorts of things will result in 2 different species not interacting. So they don't interact, so mating doesn't happen. It's sort of pre-zygotic versus post-zygotic. So pre-zygote is, of course, when we have fertilization of a sperm and egg. So these are all things that would happen before a sperm and egg fuse to form a zygote, to actually form a new member of that population or of that species. So if there isn't this separation of activity, of time of day, of where they live, there can in fact be isolation actually post-mating or during mating, where perhaps the lock and key of their genitalia don't quite work, or if there is some sort of a copulation, there is isolation where the sperm would not be able to reach the egg to fertilize it for some reason. Now, if in spite of all these sorts of prezygotic mechanisms occur, there is a copulation and fertilization, well, oftentimes the zygote might just die because it has the sperm and egg from two different species. One example is it might create a sterile hybrid. So an example of this is a horse and a mule. A horse and a donkey, both lovely creatures, different species. However, if a horse and a donkey do in fact copulate, do in fact mate, they create a mule. The mule itself is a live animal, and it survives. It doesn't die like these individuals, but it does not reproduce. A mule is sterile, so it cannot actually reproduce. So it's a dead end. So a donkey and a horse can mate, but they don't actually. They create an individual. They have a beautiful mule son or daughter, but that's it. It's a dead end. Another interesting variation is what's called F2 fitness. So this is a situation where over generations, where there might be a fertile hybrid, unlike our friend the mule, but their offspring has reduced fitness and the like. An example of this might be like a liger, which is a lion and a tiger. There is, in fact, there's a liger, which is a lion and a tiger. And then there's a tigon, which is a tiger and a lion. The first letters represent who is the male of it. So a male tiger and a female lion can reproduce. They create a tigon, which is a half tiger, half lion, which can reproduce, but their offspring will not necessarily thrive and will have reduced fitness. A liger would then be a male lion and a female tiger. So yes, those things are real. It's not just something made up by Napoleon Dynamite or whatever. These are real things. These are real animals. Usually in nature, you have habitat isolation, you've got behavioral isolation, and the like. But these sorts of things will do in fact happen. So again, these same sort of things: the habitat, the time, behavior, the mechanisms of the mechanical isolation, the separation of genitalia not functioning correctly or adequately, and the gamete isolation, and these postzygotic as well. So, an example of behavioral is courtship displays. So this here is a blue-footed booby pair, and here we have the male, and the female clearly is interested, wondering what's going on over here, but they're doing a little dance, and that dance is definitely getting her mind to thinking, I think I would like to mate with this individual. So if another bird came over and saw this blue-footed booby dancing around, they'd be like, what is this idiot doing? What the hell? What? No. But within the species, this courtship display, this little dance, this parade of available manhood or birdhood is in fact appealing and is a behavior that will isolate the blue-footed booby from other animals, from other birds. The post-zygotic isolating mechanism of mules, a horse and a donkey mate, and the result is a mule that is half horse, half donkey, and zero fertility. So, a variety of things can happen. How can this speciation happen? And there's major methods that we'll kind of talk about with how this speciation occurs. Here we have a whole bunch of salamanders in California, and each one of them has a specific geographical location. This separates them from all of the other salamanders. So in this case, they're all in the same genus, but at some point there has been separation. Maybe here you've got the Sierra Nevada mountains, so this individual is a mountainous salamander, and this one is sort of, I've been to California a while, maybe mid-Napa Valley, it likes wines, etc. Each of these individuals has geographic separation. This one right here probably lives up in the Redwood area, and the like. So each of these are all related. They're all closely related, but they have gone through a speciation because they have separation of their geography, and each one is adapted to their specific area. And this is known as allopatric, where they occur in a variety of different areas, and there is that geographic isolation. So these populations are not going to interbreed because there are characteristics of the geography of California that separate them and cause them to be separated. Sympatric population, on the other hand, is when it happens without geographic isolation. And this can often happen in plants. In this example, we have two different species of wild wheat. And normally, these two different species, when they try interbreeding, have a sterile hybrid. But what's interesting is these can, in fact, actually go into methods of polyploidy reproduction, where, we talk about this being a diploid number and this being a diploid number. If you have a situation where you get a haploid gamete from this individual with 2n as being 24, with 2n being 28, half the genetic material, the haploid number for this individual is going to be 14. This one right here, you create gametes, a haploid number, this diploid number is 14, it's going to be 7. So 7 plus 14 is 21. It will not be a successful hybrid. However, if you actually take the entire genetic information from this one and cross it and double this chromosome number, which happens within plants, you can actually have a situation where this new species occurs, this breadwheat occurs, and it has a different genetic it has a different diploid number from both of the parents and what the hybrid would be. So these are the sorts of things that happen. This sort of speciation can happen without any isolation. It is simply happening sympatrically in the same location. Probably one of the most Famous examples is, of course, the finches that Darwin learned about when looking at the finches during his studies of the Galapagos Islands and the like. And this is the idea of adaptive radiation, where you have a huge spread of different species, all from a single ancestor. We have all of these finches are related. However, each of them has its own specific location. Half of these are ground finches, so we have separation of those that were found on the ground versus those that were found in trees. And then we have separation of what type of food and what type of beaks. And ultimately, all of these differences became sort of concrete and separated because each of them had unique beak characteristics that were adaptive to their environment and began to create these separate species, all from a common ancestor, all with different characteristics adaptive to different life, different location, different food, and the like. They all may look similar, but they would not interbreed. This separation of location and of diet and the like would have ultimately resulted in formation of all these diverse new species. And that is what's known as an adaptive radiation. So why is reproductive isolation an important component of speciation? Well, if you continue to have reproductive success between the individuals of different species, that gene flow will be a preventative to actual speciation occurring. Of course, the difference between microevolution, the stuff we talked about within a population, macroevolution, the formation of new species, very different. And then, of course, we'll have sort of a combination of sympatric speciation and allopatric with our Darwin's finches, but just based on different types of separation. Okay, so the next couple of slides are dealing with the fossil record and what we've been sort of how that can help us understand information about the evolution through time. Of course, fossils allow us to understand what species lived in the past, and a paleontologist will actually study that. When we think about the history of life, 5 billion years or so, humans ourselves have only been in and around about 0.04% of the history of life. So we're relative latecomers in terms of the diversity of life that we see. Of course, we have these major eras, each kind of showing different things happening. And as we go through the diversity of plants and animals, we'll kind of go back to this idea, thinking about what exactly happened during the Paleozoic, where you have sort of the first animals. You also have these areas of mass extinction, a variety of different things. There have been 6 mass extinctions over the history of life that led to different things dying, as well as new things coming into the world. But here is the oldest. Here are the oldest sort of things. So the Earth forms 4.5 billion years ago. This is million years ago, so 4.5 billion years ago. The oldest known fossils are from 3.5 billion years ago, and these are those blue-green algae that create mats that can be found even today in Australia. But a lot of these things are kind of squishy and don't leave a lot of fossil evidence. Of course, what we see as we're kind of going up, there was in fact a mass extinction somewhere between the Ordovician and Silurian periods here, where you had a loss of more than half of the marine species. And then when that space opens up, you have a lot of speciation happening after these mass extinctions, because there's all this space, all these resources, all these things available for new things to kind of evolve and change. So thinking about sort of the first fishes occurring, and then you begin to have land plants and amphibians as we shift from aquatic to terrestrial environments. We have the shift from seaweeds to more ferns and vascular plants on land, which we'll again talk about. Continuing up, sort of the first mammals with the Triassic, the Jurassic the Jurassic period happening with, of course, the advent of all of the huge terrestrial dinosaurs, a variety of different insects, and the first small mammals appear. And again, the mass extinction, believed to be because of a meteor hitting the Yucatan Peninsula and having a profound effect on all of life. Once those larger reptiles, the dinosaurs and whatnot, go extinct, you have an amazing radiation of mammals. And ultimately, in this last period, we have some of the earlier humans, and then, of course, what we consider sort of the age of human civilization. And this is about 10,000 years ago to today. So breaking it apart, we have, again, all of these different times to think about, and a fossil record to kind of help us understand it. So when we look at that, when we think about, it's hard to think about a year, 10 years, 1,000 years, 1,000,000 years, 1,000,000,000 years. It really, when you try to think about these extensive periods of time and think about all the things that have lived and no longer live, as well as all the amazing things that live today on this earth, we don't really we have to think about, well, how fast does speciation occur? And essentially, there are two different kind of models that we like to present in terms of how this happens. One is kind of a gradualistic model. Darwin kind of supported this, showing it as a history, an evolutionary tree, and sort of a lot of transitional links as you go up this evolutionary tree. Another, perhaps more recent type of species concept is the idea of punctuated equilibrium, where you have no change of equilibrium punctuated by rapid speciation. So you have less likely to have fossils that are transitional and a lot of sudden shifts. So we've done a lot of research in the fossil record looking for either support for this gradualistic model or whether punctuated equilibrium occurs. So this cartoon of birds kind of shows exactly how this might have happened, where we have today's birds and we have a gradualistic model where we have gradual separation of these two once related organisms that have a common ancestor. Stop that. And punctuated equilibrium, where we have variation from the individual, kind of a no change, no change, where specific quick times where it happens rarely, but very dramatically, versus sort of slowly but surely. Obviously, these are kind of the questions that puzzle, and I guess most of us sort of think that it's sort of a combination. If you could kind of combine these two things, you have some organisms that are sort of slowly changing and adapting, but you also have these periods of time where there is a large amount of speciation. And a lot of times that rapid change and these little side things here are actually representative of mass extinctions that can lead to new opportunities for speciation. We have these mass extinctions, huge disappearance, large number of species, and a lot of times these famous periods that occur, or that we talked about a few slides ago, actually end. The termination of those periods are due to mass extinctions. And the mammalian extinction at the end of the Pleistocene, this is going to be when your giant sloths and your woolly mammoths and the like actually go ahead and disappear, and allowing for other organisms to kind of adapt and radiate through time. A lot of people suggest, and I think this is kind of agreed upon, that they have these 5 huge mass extinctions due to meteorites and other sorts of examples of things that happened. But we ourselves are in the middle of a 6th mass extinction that is human caused due to how we utilize the world around us, how we perhaps unknowingly take away habitat, use it for our own use. Obviously, it's important for ourselves as humans to grow food, but if we are growing food at the expense of habitat, how does that relate to the organisms that live there? We all like to drive around the forest, but that's where deer live, and it kind of interacts with life, interacts with humanity in a variety of different ways. But the conventional wisdom is that we have actually killed off so many different organisms, not just the big things, not just the black rhinos, which are hugely endangered, but a variety of other things and insects that we may not even know about. There has been so many extinctions happening that we can't even keep up with how many different sorts of species existed before humanity but no longer exist. And we bear responsibility for those changes that we've made to the earth that actually lead to that current extinction that we're in. The meteorites, as I mentioned, proposed as a primary cause of the Cretaceous extinction. That's the important one that we saw with the Yucatan Peninsula, and goodbye, our friends the dinosaurs. When we're thinking about geography, when we're thinking about speciation that occurs, one of the things that has happened through time, and one of the things that was really first being discovered and discussed during the time of Darwin, was the idea of plate tectonics. How exactly our continents are built on particular plates that are shifting and moving. And this can create, with shear, it can create shifting in sizes, it can create fault lines where earthquakes occur. That pressure can also result in the movement of magma up and release of that through volcano and the like. All these sorts of things are happening on this earth and have been happening since the beginning of early Earth. Of course, this drift, there once was anyone who kind of played with a little globe or looked at like the chunks of the different pieces of the continents as they are today, they all kind of tuck into this large. You can kind of put them all together like puzzle pieces, and that is very accurate, because essentially 250 million years ago, and thinking about going back to, I like to go back to those drawings of times and epochs and periods to think about when exactly this is happening and what is happening at that time. But you can see all of these continents, this is kind of indicating what they are today. We're all in a super island called Pangaea, and that through time there has been shifting of the continental plates. North America kind of going in this direction, South America and Australia breaking off and the like. And thinking about, again, as this drift happens, how exactly did that impact what's living there and what is potentially evolving there? We talked a lot about Australia and how they have a majority of the marsupials, your koala bears, your kangaroos, and the like, and how that separation of that during, about from 65 million years ago, that separation of that would have kind of occurred and impacted speciation occurring, and how, with all this stuff moving around, how is that going to impact the environment? What's the temperature in these different places? What's the rainfall level and the like? Just went back in time. Now we're going further back in time. Okay, so understanding these sorts of processes and thinking about them at the same time as we're thinking about speciation, the environment, and the like, really can help us understand the timeline of evolution, as well as offer kind of support for our understanding of it. So the last bit that I want to talk about tonight is the idea of systematics. This is similar to taxonomy, which is the process of identifying, naming, and classifying organisms, but it actually also has, in recent years, or recent, I guess in geological time, but recently, over the last 100 years or so, has widely integrated genetic information to help us understand the relationships between organisms. So it really is the study of this evolutionary history. Phylogeny is the idea of the evolutionary history of a group. What is it related to? How are they related? How long ago did we have this split of different species and the like? So again, we've been kind of going back to this Linnaean classification, this binomial system, this nomenclature with the genus and the species. In this case, Cirripedium acaule. Cirripedium is the genus. Acaule is the species. It's italicized. You can, especially if you're using if you use the genus once, you can then abbreviate it to just a single letter, because you kind of, if you've mentioned it before, instead of writing Cirripedium acaule over and over again, you can shorten it to just the first letter of the genus and the species when doing writing and whatnot. But this is all from Linnaeus, who was a Swedish naturalist who kind of went through the world identifying things, beginning this system, and growing, and it has grown from there. Of course, we have the taxonomic hierarchy. So this species, this isn't just a name, this is referring to a lady slipper here. We have the three different domains, the bacteria, the archaebacteria or Archaea, and the eukaryotes, those of us with nuclei. We have a couple of different kingdoms. In this case, it's a plant, phylum, class, order, family, genus, species. So each of these gets more and more specific. You can see in this class, maybe you remember monocots versus dicots. If not, we'll get back into it next week. You will never have forgotten, once again, what the difference between monocots and dicots are, but these are a grouping of plants. The Anthophyta referred to the flowering plants. As its name suggests, they're related, but not very closely related to asparagus. But more specifically, they're in the orchid family. And there's the genus and there's the species. So as you go up, you have broader and broader categories which it shares. And as you go down, you get more and more specific until you end up with a species and genus within the taxonomic hierarchy. So we're going to look at combining ancestors and lineages with common ancestors to create some sort of tree. Not really a family tree, but sort of a evolutionary tree, which kind of goes down to a common ancestor and shows lineages. Now, these things are informed by a number of different ways, especially things like DNA. But we can create phylogenetic trees from other things. Sometimes we can look at the teeth of an animal and think about how the teeth of that individual animal and teeth of related animals maybe provide some insight into how closely they're related and the like. But here we have a group, and what you can see is that the common ancestor and the relationships between a sheep, a cow, a deer, and a reindeer all reflect similarities and common ancestry in evolution. So there was some sort of hooved animal that at some point diverged, and some of the population were kind of more in the bovine and some were more in the Cervidae, the deer type group, and further separated into groups that include the reindeer and the red deer, while the bovine will kind of break apart into your sheep and your cattle. Now, if you think about the characteristics that they all share, well, they're all artiodactyls, so they all have hooves that are of 2 or 3, versus some that have just a single hoof, like a horse, and the like. So if you look at the common ancestry and the relationships between them, you know that a cow and a sheep are more closely related than a cow and a deer. And that's reflected in the classification and the phylogeny. And you can see how this basically replicates the different structures within the taxonomics family, from order to family to genus to then species. So how do we do this? Well, we use a lot of different types of data. First of all, we use morphological data. That's what I was talking about when we're looking at things like the teeth of that. The teeth of the cow was probably more similar to the teeth of the sheep than that of these deer. We do look for homologous structures. That's where, and this is something we talked about very briefly when we talked about Darwin, sort of this idea of related but of common descent. So the forelimbs are vertebrates. So we've got these sweet little arms that we can wave in the air, and there are homologous to that would be the wings of a bat, other mammals that have the same, they got the humerus, they got the ulna, they got all the bone structure, they're just in a very different shape. And our friends, the whales, might have little flippers that have those same skeletal structure. They're all homologous, they have a common descent, and in this case, all the forelimbs of the vertebrates kind of are homologous. Every once in a while, though, we do have what's called convergent evolution, where two different species acquire what appears to be a similar trait, even though they're distantly related. But they may or may not actually, in this case, they aren't due to a common ancestor. They're due to these two variant, different species adapting to the same environment. Analogous structures then are where they have the same function, but not a recent or common ancestor. So the fins of the whale and the fins of a fish may be doing sort of the same thing, but they're very much not because of common ancestry. One of the things we do for systematics is look at molecular data. So if we're looking at the primate group, we can kind of see, and again, this goes to when the divergence occurred, where the common ancestor was, and how separate and different we are from these other species in the primate group. In this case, you can look at something like ribosomal RNA will actually provide some insight into that because it changes relatively little, even though these organisms are quite diverse. So we can map that out. We can map a phylogeny using DNA data to try to identify DNA and RNA data, nucleic acid data, to identify not only how are they related, how closely related, but estimate how far long ago the separation from these different groups happened. So again, we have amongst our primate brethren, we are closest related to the chimpanzee, but then the chimpanzee and human are most closely related to the gibbon versus, say, these sorts of monkeys or the capuchin or whatnot. Now we've mentioned the three-domain system already, the bacteria, which are, of course, the prokaryotic cells that arose first. And we talked about a number of different types of bacteria. We talked about the archaea, which were also prokaryotic, but had a lot of similarities to the cyanobacteria, but also had some similarities to the eukaryotes that are so incredibly important that we know that this group of organisms evolved later and perhaps have more in common with all of the nucleated kingdoms, the plants, the fungi, animals, and the protists there. So another way of looking at this, we just have two more slides, but another way of looking at this is to think about what's called cladistics, where we actually, again, look for commonalities between species and use the data that we have to try to understand that. And by doing so, we can actually kind of build apart and see how those commonalities between different species will perhaps lead to insight in terms of evolutionary group. So basically what a cladogram does is it basically takes a group of organisms, a group of species, and separates them into outgroups and ingroups, and tries to do it with the fewest number of differences possible. And so there is the idea of these shared traits that are derived, homologous ones, as well as those that are common both in the in-group and the out-group. So what does that mean? What does that mean? So here we have a number of different organisms. We have a jawless fish, we have a jawed fish, we have a newt, which is an amphibian, we've got a snake, which is a reptile, and we have a lizard, which is a reptile. And so if we're looking at these 5 organisms, what can we say that they all have, and what similarities do they have? So in this case, all but the lancelet actually have a vertebral column. Only these 3 have lungs and a 3-chambered heart. Snake and lizards have these characteristics, including amniotic membrane in the egg and internal fertilization. In terms of bony limbs, the newt and lizard have it, but the snake do not. In terms of a long cylindrical body, the eel and snake have it. So when we're thinking about these differences and creating this cladogram that has this outgroup and then all the others that share a particular characteristic, we know that this group, we can build a cladogram because they all have this one characteristic. So they're all in this single in-group, except when we talk about having a vertebral column, then lancelet is the outgroup, and the rest of them, indicated by this large purple rectangle, all of these do. And so here we have the vertebrae. And then we have sort of, the next step would be those that have lungs and a 3-chambered heart, separating the eel. Now the eel is in the outgroup of this bunch, and we have separation of all those. And then internal fertilization and an amniotic egg sort of in here as well. So we have each of these kind of represents a large, each of these represents a large step in evolution and relationships. Now, what about the rest of this stuff? Well, maybe these sorts of things are not important in terms of separation of the snake and lizard. We need to maybe look at other data about why the snake doesn't have 4 arms. I mean, other than we then call it a lizard. In terms of convergent evolution, an eel and a snake are not closely related, but they both have a long cylindrical body because they both may live in an environment where that is the best way for them to get around. It's actually advantageous to not have these bony limbs and the like. So when we're creating this cladogram, you kind of have to separate what's important from what's not important, and think about why this leads to each of these different steps in this cladogram, the formation of these outgroups, and identifying these points which represent large steps in terms of the evolution. So this last slide kind of features the major groups of the vertebrates. So you have your mammals, your turtles represent, snakes and lizards representing some reptiles. You got some reptiles here and birds. So, based upon the information, we have separation of the mammals in terms of looking at those species that don't have hair and mammary glands versus those that do. But what's interesting is we understand that if we look at the origin of scales, epidermal scales, these are all composed, whether it's the scutes on a turtle or the scales on a lizard or a crocodile, all of these are similar. And so now we have this outgroup of those with hair and mammary glands that separates the mammals versus these that are kind of connected. And here you then have, and what's interesting though is that the hair, as well as all of these different scales, are made of the same protein, keratin. Interesting stuff. And then, because we're thinking about separating the different things, we know that both the crocodile and a bird has a gizzard, which is a structure in the mouth for breaking material down in its digestive tract. So again, we're not always going to kind of come up with the same cladistic tree. It really depends on what we focus on and what we believe to be important characteristics within these different species that we're creating. But like the phylogenies, like all these family trees, like the taxonomy, it all provides understanding and insight into how things are related to each other and how we understand our biological world. So that ends the material for Unit 3. We want you to take the exam for Unit 3 by Sunday, and keep up the good work. I'll see you all tomorrow in the laboratory. But as always, if you have any questions or if I can be of any other assistance or clarification, please do not hesitate to stick around, and we will see most of you tomorrow. Thank you.
Have a good day.